Hello everyone, in this video we will try to understand the last steps of metastasis, which is colonization. After cancer cells have successfully traveled through the bloodstream or lymphatic system, they arrive at a distant site in the body. To form a secondary tumor, these cells will colonize and must be able to adapt to the microenvironment of the new organ and establish themselves in that location. Cancer cells may undergo changes to enhance their ability to survive and proliferate in the new setting. But how do colonizing cells overcome the stromal change? For cancer cells to spread successfully, they need to create a network of blood vessels in the new location, which is vital for proper metastatic colonization. Some breast cancer cells achieve this through a process called vascular mimicry. This involves overexpressing specific genes, namely SERPINE2 and SLPI. How do cancer cells adapt and grow? When cancer cells colonize new areas, they can adapt and grow by using the body's nerve signaling pathways. In brain metastasis from breast cancer, lung cancer, and melanoma, these cells interact closely with capillaries near nerves. A protein called protocoderin 7 helps cancer cells connect with astrocytes using gap junctions composed of connexin 43. Metastatic cancer cells use these junctions to transfer the second messenger CGAMP to astrocytes, activating pathway and producing inflammatory cytokines such as IFNA and tumor necrosis factor. These molecules, in turn, activate STAT1 and NF-κB pathway in brain metastatic cells, supporting tumor growth and resistance to chemotherapy. Next, moving on to cancer cell dormancy. Dormancy can be defined as an arrest phase in cancer progression that occurs during the primary tumor formation phase or after invasion into secondary sites, known as colonization stage. Common sites harboring disseminated cells include soft tissues such as brain, lung, lymph nodes, liver, as well as calcified skeletal tissues. Next, moving on to the mechanisms that govern dormancy. Regulation of tumor cell dormancy involves the relationship between the environment and mechanisms that control transcriptional programs. The signaling cascades that play a critical role in governing cell dormancy are RASMEKERK or MAPK pathway and PI3, KAKT pathway. These pathways can be activated by a variety of extracellular stimuli, including growth factors, cytokines, insulin, environmental factors, oxidative and genotoxic stress. The activation of stress signaling pathways induces a sustained state of quiescence that is linked to dormancy. One of the examples in osteoblasts, there are several factors secreted by the pre-metastatic niche, PMN, such as mesenchymal cell-derived bone morphogenetic proteins, and growth arrest-specific 6, which also shift cancer cells towards dormancy. One of the bone morphogenetic proteins, BMP7 secreted by the PMN, activates the metastatic suppressor gene NMYC downstream regulated gene 1, leading to an increase in P38 MAPK activation, cell cycle inhibitor P21 expression, and cell cycle arrest. Molecular interactions between mitogen and stress-induced signaling are vital in regulating the dormancy state of metastatic cancer cells. The ratio of extracellular signal-regulated kinase, ERK1/2, to P38 MAPK then regulates the cell cycle. High levels of ERK1/2 activity favor proliferation, whereas high levels of P38 favor dormancy. Increased P38 MAPK activity triggers the activation of the unfolded protein response, which upregulates activating transcription factor 6, thus promoting cell arrest and survival. Moving on to how dormancy occurs in metastatic clusters. Dormancy in metastatic cancer clusters occurs when the rate of cellular proliferation within the cluster is equal to the rate of apoptosis. This balance is achieved through three factors, which are suppressive gene signaling, restricted angiogenesis, and an active immune microenvironment. The first factor, suppressive gene signaling, is achieved by the induction of differentially expressed in chondrocytes 2, DEC2, a tumor suppressor gene. The transforming growth factor beta induces chondrocytes 2, which inhibits cyclin-dependent kinase 4 and at the same time activates P27, forcing the cell to enter a state of quiescence. The second factor, restricted angiogenesis, allows blocking of blood vessel formation through activation of thrombospondin 1256 or through the inhibition of chaperones, such as heat shock 27-KDA protein, which pushes metastatic clusters into a dormant state. Lastly, active immune microenvironment is also one of the factors that govern dormancy in metastatic clusters. The immune system is a major factor in cancer anti-proliferation, in which macrophages, T cells, and NK cells clear metastatic cells through cytolysis. Next is the reactivation of cancer cells. Some of the factors are the immune-related inflammatory pathway, tissue angiogenesis, and organ-specific mechanisms. First is the immune-related inflammatory pathway. Dormant cancer cells can be reactivated with chronic infection at the site of metastasis. The neutrophil will gather at the site of metastasis and release a proteolytic enzyme, triggering the remodeling of the extracellular matrix, which activates a receptor that disturbs the ERK and P38 MAP ratio, where the P38 MAPK is inhibited, while ERK, which favors proliferation, is activated. Next is tissue angiogenesis. A low level of angiogenesis inhibitor paired with the pro-angiogenesis inducer will trigger the imbalance of the pro- and anti-angiogenesis factors, causing the angiogenetic switch, which starts the angiogenesis or neovascularization process and simultaneously reactivate the dormant cancer cell. Lastly is organ-specific mechanism, specifically osteoclast. The stimulation of the osteoclast process, such as from ovaryectomy, castration, and vitamin D deficiency, has been observed to cause reactivation of dormant cells. Both studies by Lawson and Liu showed evidence of the reactivation of dormant cancer cells when exposed to the osteoclast process. The plasticity of a cancer cell is defined as its ability to change the phenotype or traits without any genetic mutation. This causes cancer cells to have intratumor heterogeneity, and so far, there are 3 models that explain it. Firstly, the clonal evaluation model theorized that differences in the cell can be observed to be caused by random genetic or an inheritable trait alteration together with the influence of the microenvironment where the best cancer cell will be selected for proliferation. Next is the cancer stem-like cell model. Some cancer cells carry the same characteristics as stem cells, where it has the self-renewing ability and the ability to differentiate accordingly, which allows for long-term tumor growth. Lastly, the CSC plasticity model. This tumor heterogeneity cancer cells are dynamic, allowing shifting from non-CSC to CSC states. This model emphasizes the cancer cell differentiation and evolution ability, which promotes a new kind of complexity. The shifting ability allows the cell to alter accordingly in response to physiological stress and to the constraints of tumor growth. Metastasis suppressor genes are defined by their ability to inhibit metastasis in a secondary organ without affecting tumor growth at the primary site. Metastasis suppressors target pathways and proteins that are associated with cancer progression, invasion, and the establishment of metastatic colonies. In highly metastatic cells, levels of metastasis suppressors are often downregulated, the rate of gene expression lower. This downregulation is associated with the increased ability of cells to metastasize. An example of metastasis suppressor, microRNA suppresses oncogenes and inhibits signaling pathways associated with tumorigenesis. In conclusion, colonization is where cancer cells that have disseminated from the primary tumor successfully establish and grow in a distant organ. This step is pivotal for the progression of the disease as it involves the intricate interaction between the disseminated cancer cells and the microenvironment of the new tissue. Understanding the mechanisms of colonization is crucial for developing targeted interventions to disrupt this process, ultimately providing the prevention and treatment of metastatic cancer.